Okay. Well, first off, thank you everyone for inviting me to give a talk. I'm really excited. Today I'm going to talk to you about a paper that we've recently finished. It's going to be published here soon, hopefully. But it's about speciation, adaptation, and phylogenetics in the Floridian watersnakes. There we go. So speciation, as many of you probably know, is an evolutionary process by which populations evolve to become distinct species. For a long time, the most common form of speciation has been thought to be allopatric speciation, where species become geographically isolated, establish a reproductive isolation, and then this means they can no longer breed with one another, and so they are two distinct species. And to see an example of allopatric speciation, we can just look at your own coastline down there. For some reason, it's not letting me click. There we go. Your own coastline. So our beloved Bothrops insularis, the golden lancehead, became isolated to this infamous snake island through this process. But because allopatry was believed to be the most common form of speciation, phylogenetics really developed with the mindset that the tips of the phylogeny are these isolated units. So today I want to talk to you about how our understanding of speciation and phylogenetics has changed with the advent of next generation sequencing. So a phylogeny is a hypothesis regarding the relationships among a set of species or taxa. And because a phylogeny is a hypothesis, there's obviously a chance that that phylogeny is wrong. And in fact, they often are. So in the past, the most common way of estimating a phylogeny was to sequence a single gene, often a mitochondrial gene like cytochrome B. We'd build a tree with these data and then say, here's the final tree. This is definitely how these species are related to one another. And this was great. But as the cost of sequencing decreased, biologists began to discover a little bit of a problem. If we sequenced more genes, we were often recovered phylogenies that disagreed with one another. So here's another phylogeny with CO1. Here's another phylogeny with ND4, and they all have these different topologies. So now we have three different hypotheses for the evolutionary history of this group, and the question becomes, which tree is the correct tree? Now, some of this incongruence among the phylogenies can be explained by something known as incomplete lineage sorting, or ILS. And broadly, what ILS is this disagreement between the gene tree and the actual species tree. So in gray, you'll see the species tree, and the colored line inside that, and shown over here, is the gene tree. And the cause of this disagreement between the gene tree and the species tree is due to the occurrence of an ancestral polymorphism and how that polymorphism progresses through evolutionary time. So here the ancestral polymorphism makes it seem as though the green lineage diverged prior to the blue lineage, but in reality, the species divergence followed a very different pattern. So we can say that this gene failed to completely sort into its appropriate lineage. So in response to this, a concept known as the multi-species coalescent was developed, and it really helped deal with this problem. But it still didn't really solve all of our problems. Biologists were still getting a high proportion of incongruent trees, and that didn't match the expectations of incomplete lineage sorting. So they did what any biologist or phylogeneticist would do, and they sequenced more and more and more genes, thinking that eventually one tree would emerge as the most common and therefore be the correct phylogeny. And in fact, some people have sequenced the entire genome of clades. So here's an example of heliconius butterflies published recently, where they sequenced the genomes of 20 species and built phylogenies across chunks of the genome. So you can see that different parts of the genome recover these different topologies. In red, we see it recovers this phylogeny here. In blue, this phylogeny here. And even though they sequenced a full genome, there's still not really a clear winner. These two trees up here at top, they occur almost equally common to one another. So how do we deal with this, and what is causing this incongruence? Well, it turns out the tips of our trees are not quite as stagnant as we used to think. They're frequently interbreeding and hybridizing throughout their evolutionary history. For example, hybrid speciation is the concept where two ancestral lineages hybridize to form a whole new lineage. And hybrid speciation can produce a genome with nearly equally common gene trees across the genome. Similarly, introgression can cause these patterns as well, but introgression is a much more complex of movement of the genes and mixture of the parental genes due to hybridization and then back crossing of those hybrids over several generations. And introgression doesn't form a new lineage, but really complicates this idea of a bifurcating tree. So instead of having these clean branches, we now have a new branch that connects B to C as well. So hybrid speciation and introgression are very similar in that they both occur as a result of gene flow. And these processes, they really challenge the concept that species must be reproductively isolated and that allopatric speciation is the most common form of speciation. But these are now becoming more and more recognized. But speciation with gene flow is still a pretty difficult concept to grasp because we know that gene flow is a homogenizing force. If these two populations are sharing genes with one another, eventually both the red population and the blue population are going to end up being the same purple population. So how can divergence and speciation occur when there's gene flow occurring? Well, one way this might happen is ecological speciation, which relies on natural selection and local adaptation. So imagine instead of these populations existing in the same environment, which is represented by this black circle, they were instead locally adapted to their own environments. So now a blue circle and a red circle. So now if selection is strong enough, these populations can maintain their divergence or differentiation in spite of gene flow. So migration and selection sit at a balance where gene flow can muddy our phylogenetic signal, but populations are still divergent due to selection. So the objective of my study was to really test for ecological speciation, but this can be really challenging. The first thing we need to do is know whether gene flow is occurring between the two populations of interest. We also need to know whether there are ecological differences between the populations and exactly what those differences are. And the most challenging part of ecological speciation is identifying loci that are associated with the environmental difference under selection. And this is the most challenging because if the difference between these two habitats is temperature, for example, what exactly is the temperature gene that should be selected? Now, in addition to these requirements, we also have an expectation that there's going to be a pattern of isolation by environment that will occur despite a pattern of isolation by distance, or even be stronger than a pattern of isolation by distance. So in other words, populations in habitat 2 are going to be more closely related to one another than because they occur in the same habitat, than any population in habitat 1, despite these two populations being geographically closer to one another. So throughout this talk, I'm going to basically cover these points here, and test all of these requirements and expectations of ecological speciation. But like I said, this can be really difficult, and it's best to really choose a system where we know what genes are going to be targeted by the environmental selection pressures. So we're studying salinity as our ideal environmental pressure for testing ecological speciation because it's a physiologically harsh selection pressure. I don't know if any of you have ever drank seawater before, but it's not really good for you. And in fact, it's not really good for anything unless you're specifically adapted to that environment. For example, less than 1% of reptiles are known to use saltwater habitats. But what is wonderful about salinity is that its targets in the genome are pretty obvious and pretty well described. It's going to target genes that are related to osmoregulatory function and ion transport. And because our lab is really focused on snakes and reptiles, of course, we're obviously going to exploit that 1% of reptiles that use saltwater. And luckily, we can do so from our backyards. So the North American watersnakes are the ideal group for our study. This small clade of snakes, despite only 1% of reptiles using saltwater, this small clade of snakes has 4 lineages which use saltwater. Furthermore, they're really widespread and they're really abundant, which means collecting them is pretty easy. And many of the species are sympatric with one another, which means they have the potential to hybridize with one another. And in fact, we do know that many of these lineages do hybridize with one another. Now, for my study specifically, I focused on these bottom 2 species in this phylogeny. Nerodia clarki is the salt marsh snake, and it's shown over here on the left. And then Nerodia fasciata, which is the banded water snake, here on the right. Now, these two species are very closely related, and they've had a really complicated taxonomic history. Initially, they were recognized as subspecies of Nerodia cypodon, and then they were elevated to their own species, then they were sunk back into subspecies, and then re-elevated into their own species again. And the reason for this complication in their taxonomic history is the variation that we see in the morphology of these species. So this pile of snakes that you see up here, these were all collected from the exact same pond. And so you might understand why it's really hard to classify what an individual is. But it gets even more complex because within these two species, they also have their own subspecies. Within Florida, Nerodia clarki has three subspecies. Nerodia clarki, Nerodia clarki compressicata, and Nerodia clarki taeniata. And Nerodia fasciata has two subspecies, Nerodia fasciata in the north, and in the peninsula, Nerodia fasciata pict of interest. Now, the evolutionary history within this complex and among these subspecies is yet to be resolved. These species are largely considered separate, but these species are largely considered separate due to ecological characteristics. Nerodia fasciata is found in inland freshwater environments, and Nerodia clarki is generally found along the coast in these saltwater environments. And despite preferring these saltwater environments or being found in these saltwater environments, no study has been able to find any morphological adaptations to the saline environment. For example, salt glands or the structure of the kidney, none of that has been able to be found. And yet they're able to maintain an ion balance more effectively in saltwater than Nerodia fasciata. So the mechanisms of exactly how this happens is not really well known. It's been pretty elusive. And no studies have yet to test for loci under selection associated with this environmental change. Before I move on, I just want to point out that the colors in this figure, they're going to be maintained throughout the presentation. So the reddish colors are always going to be Nerodia clarki, and the bluish colors are always going to be Nerodia fasciata. So our hypotheses, given the ecologically divergent nature within this complex, we hypothesize that the adaptations to saline environments are able to resist the homogenizing effects of gene flow between Nerodia fasciata and Nerodia clarki. And if adaptation facilitates differentiation in spite of gene flow, we would expect to find genetic structure corresponding to environmental differences between Nerodia fasciata and Nerodia clarki. Now, being phylogeneticists, we were really initially interested in the general phylogenetic structure of this complex. So the first thing we did, of course, was sequence a single mitochondrial gene, specifically cytochrome B, for several individuals. And over here on the right, you'll see the resulting phylogeny. The two columns of color represent the species and subspecies assignments when we captured the snakes. And what you'll see in this phylogeny is that there's not a whole lot of phylogenetic signal. Most of these are polytomies, which means there's not really any clear clades. And the only real split that we see in this phylogeny is between the mainland and peninsular Florida, right about here. And this is to be expected based on the geographic history of this region. Specifically, this is a really common biogeographic break seen in many other taxa, including plants, salamanders, other snakes. And this break is caused by Pleistocene sea levels, which separated the peninsula from the mainland. But we still don't have a lot of resolution otherwise, and we weren't really satisfied with this, so we did what any good phylogeneticist does and sequenced more data. But even with these several more additional mitochondrial genes, several more nuclear genes, we still found little to no signal between the species or subspecies. And again, the only divide we really see is the mainland peninsular split. So we decided that single gene sequencing simply wasn't going to cut it, and we moved on to next generation sequencing. Specifically, we identified SNPs using a protocol known as double digest restriction site associated DNA sequencing, or DDRAD-seq for short. So this technique chops the genome into lots of pieces using restriction enzymes and sequences around these cut sites. And we did this for 118 individuals and ended up identifying 7,057 SNPs. So this is a lot more data. Hopefully now we can actually see some phylogenetic structure or see some structure, some genetic structure in general in our complex. So the first thing we did with these data is an analysis called DAPC, which stands for a discriminant analysis of principal components. And this analysis is blind to R species assignment. DAPC is just looking at the genetic data and trying to maximize the separation between groups while minimizing variation within groups. So we ran several different models for different numbers of potential populations that we could have in our data, 1 through 20. And what this is showing is that at 5 or 6 populations is probably the most likely number of populations in our data. So we can take the assignments that the DAPC gives to an individual, and we can plot those on a map. And this is what it looks like at k equals 5, so at having 5 populations. So at k equals 5, the populations or genetic clusters, they match really closely to our expectations of the subspecies distribution. This was pretty surprising to us. We actually weren't expecting this, but this is really exciting. So we finally have a data set that's able to parse out some genetic structure, and that genetic structure matches our expectations. And although the DAPC assignments are blind to our species assignment when we capture the snake, it actually agrees with our assignment 80 to 90% of the time. So we did a pretty good job in the field of identifying these things, surprisingly. At k equals 6, the only population that's impacted is actually Nerodia clarki compressicata, which is the subspecies of clarki found along the coast. And what happens at k equals 6 is that the southern portion of the range is actually split from the central portion of the range. And although k equals 2 was not our best model by any means, I wanted to plot it because we have 2 species, hypothetical species here. So I wanted to know what the data looked like at k equals 2. And it similarly shows this coastal inland dichotomy that we saw at the k equals 5 and k equals 6 assignments as well. So in the remaining analyses, instead of using our species assignment at capture, I'm going to be using these DAPC clusters as our subspecies and species assignment. So at k equals 2, these are our Nerodia fasciata individuals in blue, and at k and in red are the Nerodia clarki individuals, and then so on for the k equals 5 and k equals 6 populations. Now, after DAPC, I specifically began to wonder if these clusters, because again, I didn't really expect to find these subspecies clusters. So I began to wonder if this could just be due to sampling bias. A lot of the samples up here, these were our Nerodia clarki taeniata individuals. They were collected from really close ponds. So could it just be that isolation by distance is influencing our data? That things that are closer together are just clustering together because they're more similar to one another. So to answer this, I ran a analysis called construct. And construct is similar to DAPC in that it is a population clustering method, but it's really neat because it can account for the pattern of isolation by distance and ask how many significantly different isolation by distance clines are there. So again, we ran several models for different numbers of populations. And we also ran 2 different kinds of models: spatial models, which accounted for isolation by distance, and non-spatial models, which did not. And what you're seeing here is that spatial models always perform better. And so this means that isolation by distance is definitely impacting our data. The other thing this graph is showing is that after k equals 2, after 1 additional layer, we're not really getting a whole lot of additional resolution. And this is confirmed when we actually look at the contributions from each additional layer. It shows that at k equals 2, this layer contributes quite a bit, but after that, it contributes very little to the overall genetic structure. So now we can take these assignments and plot those again on our map. And again, we see this coastal inland divide. So definitely 2 species here, but our subspecies have now kind of dissolved due to isolation by distance. The other thing that we can see in this analysis is quite a bit of what Construct calls admixture. Now, this admixture could be due to gene flow, or it could be due simply to shared ancestry. But what we see is that the individuals identified as Nerodia clarki tend to have quite a bit of signal still from Nerodia fasciata. And this, again, could be due to just shared ancestry. They were recently diverged, and so the signal just hasn't been lost from Nerodia fasciata yet. Or perhaps there is gene flow occurring from fasciata into clarki, or just between the two species. Before I tested for gene flow, I did want to see what the phylogenetic structure of these clusters were, our genetic clusters were. So I used a program called SVD Quartets. And on the right, you'll see the phylogeny for all 118 individuals. And on the left, the species tree for our DAPC clusters at k equals 5. So the bars on the right of the phylogeny represent the construct admixture, that same plot that I just showed you, as well as the DAPC cluster assignments at k equals 2, 5, 6, and 7. So the first thing you might notice is that our clusters generally form these easily identifiable clades. So we have Nerodia fasciata up here, Nerodia fasciata pictiventris here, Nerodia clarki taeniata forms its own clade here, and then the southern compressicata and Nerodia clarki clade as well. However, if you know anything about phylogenetics, you'll also see maybe some weird things. So first, this species tree places Nerodia clarki taeniata sister to compressicata, where in this tree over here, clarki actually sits closer towards the end of the tree, not really the right terminology there, but sits sister to compressicata and instead. Second, many systematists prefer that species are monophyletic, but you'll see that this is not the case for Nerodia fasciata pictiventris. It forms a paraphyletic clade. And the other thing is that Nerodia clarki compressicata is also heavily paraphyletic as well, actually forming two distinct parts of the tree. And this paraphyly is likely due to the low support that we see here at the base of the tree. And this could be due to gene flow, and we kind of expected it to be due to gene flow. And that's what I set out to test next. So using our DAPC k equals 5 populations, we used the program TreeMix to infer gene flow, specifically introgression among our populations. So TreeMix identified a pretty large amount of introgression occurring from fasciata into Nerodia clarki on several different points of the tree. We also tested for hybrid formation using a program called HYDE. And HYDE identified that some of these lineages may actually be the result of hybrid formation, where ancestral Nerodia clarki and ancestral Nerodia fasciata actually came together to form these new lineages. And this is particularly interesting because previous hypotheses have suggested that Nerodia clarki taeniata is derived from Nerodia clarki, and these two subspecies are actually pretty geographically distant from one another. So this hybridization could have occurred sometime in the Pleistocene when sea levels were high, and then formed this new Nerodia clarki taeniata lineage. This analysis also suggested that Nerodia fasciata pictiventris may be a hybrid between fasciata and clarki lineages. And if you remember, Nerodia clarki pictiventris was paraphyletic. Nerodia fasciata was paraphyletic due to the placement of Nerodia fasciata pictiventris. And so this gene flow or this hybrid formation might help explain why that paraphyletic placement occurred. But what I found most interesting was this directional gene flow from Nerodia fasciata into Nerodia clarki. The next thing we wanted to do is just visualize what this gene flow looks like. So we used a program called EAMS, which stands for Estimating Effective Migration Surfaces. And we see quite a bit of, we see high gene flow within the inland population. High gene flow is showed by blue. And high gene flow along the coasts as well. but a reduction in gene flow between the inland and coast. However, there are several pockets where gene flow might be occurring, specifically here in the central part of the range. And if you remember from the phylogeny again, Neurodia fasciata, or Neurodia clarki compressicata, which is found in the central part of the range, was heavily paraphyletic. So again, this high admixture zones may be what is causing that high paraphyly in Neurodia clarki compressicata. So at this point, hopefully I've convinced you that there is a high prevalence of gene flow between these lineages. So we can go ahead and check our first box. The next thing we wanted to do is identify ecological or niche divergence between these species. So to do this, we attempted to use a program called Humboldt, which measures niche divergence and niche overlap. But unfortunately, the results of Humboldt were inconclusive. And this is because Humboldt and any other niche analyses require you to collect background data. And when you have species like Neurodia clarki and Neurodia fasciata, which are butt up against the coast of Florida, there really is no background data to collect. So this was unfortunate, but what Humboldt did do is identify the most important environmental variables that separated these two species. And many of these variables made intuitive sense, such as the amount of flooded vegetation and temperature. But there's one variable that Humboldt identified that really stuck out to us. And I'm going to jump into that variable specifically. Now, unfortunately, there is no data available for salinity specifically, no environmental salinity data. But this variable that stuck out to us is highly correlated with salinity. And this is soil cation exchange capacity. And we can see that between the two species, Neurodia clarki and fasciata, that they are highly divergent or highly different. But what's really interesting is when we look at this data alongside our construct admixtures, we actually see a pretty tight correlation where individuals which share the most ancestry with Neurodia fasciata over here on the right side of the graph, they also occupy the least saline environments. And individuals that share the least ancestry with Neurodia fasciata, or the most divergent from Neurodia fasciata, they occupy the highest salinity environments. So in other words, salinity may be acting as a barrier to gene flow. So we can go ahead and check off another box. This is ecological divergence. Now, before I go into testing those pesky loci under selection, I want to skip down to this expectation of isolation by environment. We now have the data necessary to test this expectation, and we can do so using Mantel tests. So the first thing we wanted to do is test if genetic differentiation is associated with geographic distance. In other words, is isolation by distance occurring? And we already have an idea that this is occurring due to our construct analysis. We already showed that isolation by distance is occurring. And when we run the Mantel test, again, this is exactly what we see. We see significant isolation by distance pattern, both across our species, and if we look within a single species, we also see a pattern of isolation by distance. Where it gets interesting is if we test if genetic differentiation is associated with environmental distance, or the amount of distance in salinity. And we can control for this pattern, this known pattern of isolation by distance. So here we should expect no signal within a species. So within Neurodia fasciata and Neurodia clarki, there should be no significant isolation by environment pattern occurring because they occur in the same environment. But when we look across our individuals, we should see a significant pattern, and that is exactly what we see. So we can say that isolation by environment plays a large role in the divergence of these two lineages, but not the subspecies within those lineages. So we can check that box as well. The last thing we need to do is look for those loci under selection. So because we're dealing with salinity, the genes that are under selection, again, should be those associated with ion transport and osmoregulatory function. So to test this hypothesis, we ran a program called Bayescan to look for outlier loci between our two species and found 41 loci of interest. We then took these 41 loci, we blasted them to the Thamnophis elegans genome, which is the closest relative genome that we have. And we extracted genes which fell within 2 kilobases of a blast match. And this led to 31 potential genes of interest. We then can take those genes and identify gene ontology or Go terms that are associated with them. And we can use a program called ShinyGo to identify Go terms that show up over and over again, or are what are called significantly enriched. And we identified 24 significantly enriched Go terms. So let's take a look at what those look like. So most of those Go terms are related to osmoregulatory function. So on the left, you'll see a network of significantly enriched Go terms and how they're connected to one another through the number of genes that they share with one another. On the right, you'll see those genes and the Go terms with which they're associated. And looking at the list of Go terms, which are these numbers here in the network and this plot here, you'll identify a pattern pretty quickly. You'll see terms like cation transport, ion transport, metal ion transport. And if you look back at the genes, you'll also see genes that have been previously associated with adaptations to salinity, particularly these SLC genes, which are solute carrier genes or proteins. So we can check that last box there and say that we have pretty solid evidence that ecological speciation is occurring or driving at least divergence within this group. Since we have gene flow, ecological divergence, loci under selection associated with that ecological divergence, and a pattern of isolation by environment. So overall, we found pervasive gene flow and environmental selection driven by salinity is shaping the evolutionary history of this complex. The previous conflicts in phylogenetic signal that we see are likely caused by that gene flow and hybridization. We also noticed that the gene flow is asymmetrical, occurring from Neurodia fasciata into Neurodia clarki. And this could be due to one or two reasons. The first one being a source sink dynamic, where fasciata acts as a source population. It's a much larger population, and it is feeding individuals into the coast, which acts as a sink population. The other dynamic that might be occurring is a generalist-specialist dynamic, where Neurodia fasciata, again, the larger population, is acting as a generalist and is able to just move wherever it wants, while Neurodia clarki acts as a specialist and focuses entirely on the coast. Overall, though, ecology and selection for osmoregulatory function is likely contributing to the maintenance of genetic divergence in this complex. We identified genes which have previously been identified as responders to salinity stress. And so our next steps, we're really interested if these patterns, if parallel patterns can be found in the other species of Neurodia. And that's actually where a master student in our lab is going to be picking up. Overall, I hope I showed you that speciation with gene flow is a lot more common than we previously thought. We can no longer really rely on these strictly bifurcating trees. We know that gene flow is occurring throughout the evolutionary history of lineages. And the drivers of divergence may not only be allopatry, but there may be selection pressures that actually drive divergence as well. So hopefully we start to move away from these bifurcating phylogenies, and we start to focus a lot more on the idea of phylogenetic networks. With that, I'd like to acknowledge all these people, specifically my co-authors. Greg and Jason were also master students in our lab that worked heavily on this project, and then our funding agencies and helpers. And with that, I'd be happy to take any questions.
Nice.
Thank you.
Virtual claps.
Yeah.
This is better than Zoom though, because on Zoom people, I don't know if how much you guys use Zoom, but there's just like a button with hands together, and so people just hit the button and a sticker pops up. So it's not even actual claps.
That's lame. That's lame.
Yeah.
I know.
Yeah.
It is lame, but I'm also guilty of doing it myself.
We all are. Right? We all are.
So I don't know if somebody has questions for Rhett.
Actually, I had a question, but you kind of answered it on the end because, first of all, like hi, I didn't say hi, right? I forgot, man. I have no respect at all.
Yeah, I didn't say hi either, so it's okay.
Great talk, Fred. I think it's really interesting. First of all,
thank you.
And I was really struck by that asymmetry between the gene flow patterns between the two species. And I was like, dude, for me, it made no sense that the only species that was like a mixture of the two was the one on the coast, Clarkey, right?
Yep.
Okay.
While Fasciata remains like almost without any signal from Clarkey.
Yeah.
You just see a Fasciata signal within Clarkey, but not the other way around. So
yeah,
like to me that was like, okay, so it's not gene flow, because gene flow should be both ways. But then you said that it may be acting like a source sync population with only individuals from Fasciata going to the coast and not individuals from Clarkey. So like, that was the only question that I had, and you kind of answered it. So
yeah, it is a very counterintuitive pattern.
Yeah.
When we first got it as well, we were also pretty confused by it, because if they are really adapted to the coast, like how is Fasciata infiltrating that environment? Like how are they able to deal with salinity? And it's possible that they're not able to deal with salinity. They're just making it into the environment enough to breed with Clarkii and then dying off or being selected against. Yeah. But yeah,
if I can just like jump in and add like another question to that one. If you guys try to see a difference between this unidirectional gene flow using mitochondrial versus your red sac data, if you have like some kind of different signal from different data sets, that will like maybe help to understand if it's like driven by males that can copulate with the females of the other species, or vice versa, or something like that.
Yeah, that's not something that we've tested yet, but that is something that we're interested in, and I'm hoping that our new master student will kind of pick that up a little bit more. He's going to be probably sequencing, well, he's going to be trying to fill out the rest of the phylogeny and kind of get a Nerodia phylogeny, but he's hoping to also kind of sequence coastal and inland populations of all these things, and sequence not just DD rad-seq data. He's hoping to kind of expand that and do kind of low coverage genomes to hopefully be able to do a lot more kind of demographic analyses to kind of understand how this gene flow might be occurring, like whether it's females going into one environment or whatever. But that is not something that we've looked into. But it's definitely possible that I don't know if our current mitochondrial data has the signal that we need, but I don't know.
Yeah, maybe you need more genes, like
there's
only 4.
So hopefully Tristan, our new master student, will be able to sequence like the whole mitochondrial genome. I mean, if we're sequencing low coverage genomes, we should be able to assemble the whole mitochondrial genome and hopefully get
that
different.
Would be cool.
Yeah.
Other questions?
Yeah. I have a question too.
Go on.
Can I go, Juan?
Yeah. Go.
So I didn't understand how did you get to those genes that you found related to the salinity. So you show 41 loci.
Yep.
I missed the start of the.
Yeah. So we basically took our 2 populations, the DAPC 2 clusters, the Clarkii and Fasciata.
Can you go back to that slide?
Yeah.
So maybe with the slide.
Yeah. Yeah, so this is the main description. So we ran the program Bayescan, which is a FST outlier, essentially. And so it's identifying these 41 loci. There are actually, or 41 SNPs, essentially, 41 loci, that were significantly divergent among the two populations. And actually, if you look at the allele frequencies, they're actually fixed for the two populations. So all of the Fasciata individuals have one allele, all of the Clarki individuals have another allele.
And this is over the whole genome.
Yeah, that's over the...
I think, Rhett, that maybe it's a good idea to explain a little better the RAD-seq data set, because it's not like the complete
no, I see.
sequence.
It's not the whole genome. I mean, but it's not selected loci. It's selected just by the digestion of the RAD-seq.
Yeah, right? It's just the SNPs. The loci is just the data set is a bunch of SNPs.
Yep.
Okay, but so SNPs retrieved from the whole genome, independent on from what part, or right? It's not. It's
entirely correct. Yeah, it's entirely dependent on where the restriction enzymes cut, and so it's just random SNPs from throughout the genome.
Mm-hmm.
And so we had 7,057. So we just wanted to know which of those SNPs were the most divergent between our two populations, and what do those SNPs match to, essentially. What genes do they match to? So we took those 41 outlier loci, blasted them to the Thamnophis, and figured out what genes they were closest to, what genes were within a linkage, essentially within linkage of those SNPs.
Okay.
And then we extracted those genes, and then we used those genes to kind of match those to the gene ontology terms, or the function.
Mm-hmm.
Does that make sense?
And okay, but why you are like missing loci, like 41 outlier loci, 31 that you blast, and 24?
So there were, so they are, they're not going to be essentially a one-to-one relationship. So some of the things that we blasted, they didn't match any genes. They blasted to some random location in the genome, but there were no genes within 2 kilobases. So those genes, essentially, that's how it reduces from 41 to 31. Yeah.
So 31 SNPs near any gene, near a given gene.
Yeah.
Okay. And then how it goes to the 24? Oh, it's 24 GO terms within these 31 genes.
Yeah. So each of those genes has a set of gene ontology terms associated with them, a large set of gene ontology terms associated with them. And so this analysis is really looking for what gene ontology terms are overrepresented in our data, which gene ontology terms show up over and over again across all of our genes.
But but this is overrepresented compared to what? To the whole genome, or
compared to the other gene ontology terms that are in the database.
Okay.
So it's going to return 31 genes. Each of those genes is going to have a list of gene ontology terms.
Mm-hmm.
And so some of those genes are going to have unique gene ontology terms, and some of them are going to have shared ontology terms. So it's looking for those ontology terms that are shared across all of our genes.
Okay. And okay, go to the next one. And so from these 24 classes, you found most of them are related to salinity.
Yeah, ion transport across the cellular membrane. So ion transport, cation transport, metal ion transport. So many of these are just ion transport, inorganic ion transport. Just over and over again, these highly divergent loci appear to be associated with ion transport, which is what is necessary for salinity, is to transport ions essentially out of the cell.
But besides these 44 that 24 that are related to ion transport. Are there other categories that were enriched in this data set? Or is
so these
perfect like that, or only the ion transporters are the enriched?
So essentially only the ion transport ones are the ones that are enriched.
Okay.
So we have a couple
what
here.
I was trying to understand if it's driven towards this function, or it's just the result that
it is
like
just
that.
the result. So you'll see these nodules or these modules up here that are kind of isolated from the rest. These are gene ontology terms that aren't necessarily associated with ion transport. So number 3 is regulation of localization. I don't even know what that means, but it's not it's not ion transport. And then 22 down here, this has to do with exocytosis. So these are not, that's not an ion transport related function either. But they also popped up as significantly enriched. It just happens that most of the gene ontology terms, this is just how the results turned out, most of the gene ontology terms are related to ion transport.
Oh, that's really nice.
Yeah.
Yeah.
And so I don't know if you try or just test, because you test between these two, like the Faciera, I will try to pronunciate in English, and Clarkii. And so you are just like testing between these two, but I don't know if you guys try to test between the subspecies, like just to see if these in some way are not like the most variable genes. I don't know why, like just like a really silly null hypothesis. Like, okay,
no,
let's do some control here.
Yeah, good point.
You're 100% right that I probably should have done that, but I did not. Yeah, but you're correct. Like that probably would be a good thing, a good control group essentially to do this on.
Yeah, no, but it's too perfect. Like you know, but probably is correct. So really nice.
Yeah.
Other questions? One.
Hi. It's not a question really. It's more of like, I don't know, a thought I have. I have no idea if it's, I don't know. So wait, what if you, because I mean, salinity, it's not the same throughout the year, right? So it has to, sometimes it's more saline, sometimes it's less saline, right?
Yep.
So what if you approach that question of the amount of individuals from Faciata that go to the coast and may breed with Clarkii individuals by sampling? Like if you know that time of the year when saline concentration is lower, that theoretically will allow for Faciata individuals to move more easily to the coast.
Yep.
While in those times of the year that where salinity is higher, you would expect to see less individuals from Faciata in the coast.
No, that's a really good idea, and that's definitely a limitation of our study. Is like we used this metric cation, soil cation, essentially as a metric of salinity. We don't have actual salinity data, so it'd be really good to get actual salinity data and try to kind of control for the timing and the changes in salinity. So that's a really good idea. Unfortunately, we just don't have the data to do it right now.
Yeah.
So a question related to that. I don't know. Yeah, I know nothing about the geography of the peninsula, and I just know what I saw with you guys in the field, and but you can maybe like create a layer of resistance, not only like a layer based on one variable, but you can also like use several others and test them if you see the same thing. I'm just trying to like wonder if there is other things happening in the same area that can be like a confounding factor, like something that you are not taking into account that's happening in the same region? Yeah. I know that you guys, you try to use the Humboldt, you use the Humboldt to get the most important variables.
Yep.
But you only try using, only test using the, like a proxy for salinity, or you use the other ones?
So
I'm saying like in the Mantel test.
So for the Mantel test, we only tested salinity, correct? Yeah.
Yeah.
But it is possible that so we see a lot of the same patterns that we see. So this is the salinity difference. These are the other most important variables. And we see a lot of the kind of same divergence. Regularly flooded vegetation could also be influencing it. This is just vegetation that's being flooded more regularly than the inland, so it's going to be higher. So I mean, that could also influence it. There's also, you know, differences in the tree cover, the amount of water just in general that there is. There's going to be more water on the coast. I don't know. And then this is the temperature of the warmest month. So I don't know if that one influences anything, but that's but there's definitely other environmental factors that are underlie this difference between the coast and the inland. We just chose to focus on salinity because that was our kind of hypothesis.
And they prey on the same stuff?
Yeah, pretty much.
Okay, there is no difference. Okay.
Yeah.
And so the data set that you use is like it's a kind of a standard data set for ecological variables, or you build your own?
No, these are
preferred set of variables.
I took several different data sets, but they're all publicly available, like global data sets essentially. So there's the cation one came from a soil data set. The other ones here came from a land cover data set, essentially, what kind of habitats there are. And then the last one here, this is the standard bioclim layers that are used most frequently. I included those as well. The only one that came out from the bioclim layers is this temperature at the warmest month. But yeah, they were all included to try to pick out what was the most divergent between them.
Nice.
But yeah, there's definitely other environmental factors that could be influencing this. And that's really hard to parse out exactly. Yeah.
Yeah. It's a kind of an open area, like for other studies.
Yep.
Like just like to increment the IBE test.
Yeah.
Cool.
So just curiosity, where does these snakes live? Like they go to open water, they stay in the open water?
They're pretty much exclusively found. You mean well, they're all found definitely around water. So the fasciata are generally found along creeks and rivers. The Rhodia clarki, the salt marsh snakes, they're generally found in. They kind of have different. The subspecies are represented by different habitats. So the compressicata is the mangrove salt marsh snake. So it's typically found in and among mangroves, but it's in these areas that have kind of tidal influences, essentially. So their salinity increases and then decreases with the tides. And the same is true for the other clarki subspecies as well.
But do they go to the sea, to open sea?
Or not
just stay there?
Yeah, they pretty much stay in these salt marsh habitats. So they are, I don't think one has been observed in the ocean, but it also would not surprise me if they can cross the ocean.
Mm-hmm.
Yeah, I collect one in the Cascade Park.
Oh yeah,
in Tallahassee. Yeah, they are pretty common.
Yeah, they're definitely very common.
Yeah,
They're a dime a dozen in Florida.
In the Everglades, I think that Roberta was like holding one that was almost her size.
Yeah, they're smelly snakes too.
Oh yeah. More questions? If somebody has like a question and wants to type, feel free to type in the chat. I don't know, somebody can be like a little afraid to say something in English, maybe. Don't need to be, but if somebody is, can just like type the question. So, more questions? I have one. Why there is no divergence time estimation in this paper?
Because one, the fossils are not very well described, and the number of outgroups that we have are also not very large. We have a single Thamnophis sortalis as our outgroup. Yeah, and I don't. Yeah, well, we're hoping to.
Excuse.
Yeah, we're hoping to include some divergence time estimation with Tristan's project, but yeah, I wanted to, but it just seemed too challenging for the RADSEG data set.
Oh yeah, for that data set it is. It's just like to have a clue about the divergence between these two species and the subspecies, yeah, and an idea of how recent this differentiation is happening, and this kind of adaptation is happening. So how fast the rate of adaptation
yeah, no, that's
in the species is happening. That would be really cool.
That's definitely the kind of main interest in Tristan's thesis project, is to kind of get a divergence time estimation for this complex and for the whole Neurodia kind of phylogeny as a whole. And we're gonna also, like I showed you Florida, but Clarkii ranges all the way to Texas. Fasciata ranges all the way to Texas or Mississippi. So we're still missing a lot of this complex. And so he's hoping to expand the sampling a little bit more to kind of get a better idea of how the evolution has progressed in this, or how the evolution occurred in this complex.
Nice.
Mm-hmm. Rhett, yes? Are those the actual patterns of the snakes? That's cool.
Yeah, this is the general patterns of the snakes. I made this.
You
trying to get that? Yeah, I'm trying to get that MB and E cover, man.
Oh, I mean, if it were up to me, you'd get it. Definitely, it's really cool.
We'll see, but yeah, these are the general patterns of the snakes. So this, and it's kind of Neurodia fasciata is on this side, and then Neurodia clarkii are these three over here, with Clarkii compressicata, and then Clarkii taeniata here in the middle. And this is fasciata pict of interest. And I got these little fish down here to represent the direction of gene flow as well.
So creative.
I know.
Science, dude. Glow will be an artist, you know. Yeah.
It's much better than our round three in that paper. And I think we didn't get the cover.
Yeah.
They are charging me now and didn't say anything about the cover, so
charging me. Yeah. That's a discount, a 20% discount for a cover. None of that. Okay.
Yeah. Well, we'll see.
Okay. We don't have any more questions, so I'd like to thank Rhett for coming, for spending this time explaining your work. I didn't get this is your master work?
No, this is just a random side project.
Okay, a side project. Yeah.
So this project has been in the lab for like 10 years now, and no one's ever published on it. And so after the third master's student failed to actually publish it, I was like, I'm gonna finish this.
Okay.
Something's gonna come out of it. But yeah, this has nothing to do with my dissertation or thesis.
Yeah, I was wondering where this come from in your life.
The dissertation was on turtles, tortoise. Or turtle.
What's that?
Your dissertation, like your thesis, your master thesis was.
Yeah.
On
Yeah. It was on gopher tortoises.
Tortoise.
Yeah.
Okay, thank you again.
No problem. It was good seeing all of you. I miss you guys.
Thank you, Red.